Hello students, today I am going to cover a very important unit of our body, cell, the fundamental unit of life. Before we go in detail on the topic, there is an interesting story behind the discovery of the cell and the equipment which is used to examine it. While examining the slice of the cork, Robert Hooke found that it is similar to the structure of the honeycomb. That is, many little compartments are present. Cork is a substance which is obtained from the bark of the tree. This significant discovery was observed by the Robert Hooke in the year 1665, and it is by self-designed equipment called microscope. Cell is derived from the Latin word means little room. It sounds very small and insignificant, but it is very important in the history of science. Today, we will be going to cover the unit of life, that is cell, cell structure, cell organelles, microscope and preparation of slide. All organisms are made up of cell. It might be the single cell, it might be many cell. We term single cell as an unicellular organism, for example, bacteria, chlamydomonas, amoeba, euglena, etc. In plant and animals, they are made up of many cells and we term it as a multicellular organism. As I told you, all organisms are made up of cell and this cell cannot be observed through our naked eyes, and we need a special equipment which is called as a microscope, where the extra magnifying lenses are present, which is now available in the form of compound microscope. Today we will be going to cover cell structure, cell organelles. Later we will be covering on microscope, its parts and the preparation of slide. So coming back to the cell structure and cell parts. Cell structure. In plant, there is an extra cell wall present, rigid cell wall, which is made up of cellulose, which is giving a strength, extra strength, stability to maintain the structure of an plant cell. Cytoplasma. It is a jelly-like semi-fluid which is filling in between the plasma membrane and the nucleus. Little tiny inclusions are present which we call it as a cell organelles. The organelles are vacuoles, mitochondria, which we call it powerhouse of the cell, endoplasmic reticulum, where it is two types, rough endoplasmic reticulum and smooth endoplasmic reticulum, Golgi apparatus, lysozymes, ribozymes. It is called as protein factory, where all the protein synthesis is taking place. Plastid. In green plant we call it as a green plastid or chloroplast. Cilium, flagella, which is an locomotory organs. Apart from this, nucleus is also present. These are the cell structure and different cell parts. Coming to the microscope, as I told, all the organism is made up of cell, and to observe the cell it is not possible through our naked eyes, and we need a device or an equipment which is called as a compound microscope. Basically, there are two types of microscope, simple microscope and compound microscope. Simple microscope are the one which is used for the dissection sample. Compound microscope are the one which is used for the observation of the microorganism and live microorganism also. Coming to the different parts of compound microscope. Body tube, eyepiece, revolving nose piece, stage, mirror, condenser, coarse adjustment, fine adjustment and base. These are the different parts of microscope. With this, the different magnifying lenses are present. Most of the school and colleges are using a compound microscope, which will be having a magnifying lenses of 10x, 45x and 100x oil immersion objective. Next, coming to the preparation of slides. Preparation of slides is a very important step we have to follow in the laboratory practices. One, to observe it under the microscope. Preparation of slide. Take a clean glass slide, hold it to a flame, sterilize, smear with a sample which is present or given. You can take a pond water or any of the organism sample, air dry, heat fix, and different series of stains are used. For example, there are different staining techniques: gram staining, negative staining, simple staining, where we use a stain to observe or for the visibility of an microorganism. Or you can go for directly taking a pond water, put it on a glass slide, observe it under a microscope, and you will be observing different live microorganism moving there. There is another technique called as an hanging drop, where we go for checking the motility of an bacteria. It is very simple. Taking the sample, putting a drop of sample to the slide, making the drop to hang, and we are observing under the microscope, basically the 10x magnification, where you can see the motility of an organism. So cell is a vast topic. I hope I have covered whatever is required for the grade from 8 to 10th, and thank you.